That actually segues perfectly to my next question. What are common quality control failures in peptide synthesis or biologic fermentation for GLP-1 analogs, and how do we prevent them?
Deepa, do you want to go? I feel like I'm talking too much over everybody.
It's okay. You go ahead and I'll piggyback on that one.
Yeah, so I think that some of the failures that we obviously are most concerned about would be sterility, at least for the injectables. At Kylera, we have an oral program and we have an injectable program. So I can kind of give two different answers depending on which asset we're talking about. But really, the biggest thing that we have to be concerned with, and where there's probably the most danger, is to have a sterility breach. And so that's one of the things that we obviously keep an exceedingly close eye on, is the bacterial endotoxin levels as well as the sterility assurance. So when we do have a failure in those regards, it can be catastrophic. You can lose an entire batch. And certainly we want to catch that early. So I think we place a lot of importance on compliance with the Annex 1 changes that have taken place over the last few years to make sure that we try and take the human component out of the process of aseptic manufacturing as much as possible, because we can't terminally sterilize these peptides. So we have to be very, very clean in the manufacturing process, literally from start to finish. And then the other things that come out is that these peptides, they get a slight little change in the conformation can make it look very different on an HPLC assay. And so we have to really develop super solid analytical methodologies to make sure that we're accurately characterizing the impurity profile for these molecules. And I think that's really where one of the failures can come in, is people that don't put enough time into the analytical method development to appropriately pull out some of the impurities that you might see. And so you can end up getting surprised sometimes if a peak shows up underneath a main peak that you didn't know about before or expect. It can introduce a whole slew of different problems, all the way going back to your initial preclinical toxicology work, where you would want to qualify the impurities that are in your product. So I think those are probably the two big ones for us, is making sure that impurity profile is accurately put together with appropriate analytical methods, and then that the sterility assurance is there for the injectables.
So I'll add to that one. Thank you, Doug, for bringing that up. I think I'll add to that. We really need to spend more time in the beginning when you are developing your processes to really understand the process and the characteristic of the molecule before we get into the manufacturing. I think once you know what we are looking for, then it's easier to identify during the process. I think we need to spend, like you brought up, we need to spend more time on developing the right methodology. Nowadays, the technology is fairly advanced. Make a good use of it. Spend some time. If you see issues around, if you see OOS, spend some time on the investigation trying to understand what exactly has happened, versus trying to just blame it on the machine or on the manpower, and then move away, because often, this is the easiest way. Blame the analyst, blame the machine, and say close investigation. But nobody really spends time to really get to know what is happening. So I think it's in the early stage when you are developing this, your product, that's the time to make sure you spend some time, truly understand the process and your molecule, or, before you get into the next phase, because that really helps in the future.
Now, I want to get into quality control a little bit, and circle back a little bit deeper to your point earlier about batch-to-batch consistency and how we ensure that. Can you elaborate a little bit? What are we investing enough in analytical methods to ensure that?
I think that's the question to ask. Are we really investing enough? There's always a rush for everything. There's always a push from upper management to make sure that you're releasing the batch quickly, you're getting to the answers quickly. That analytical team doesn't really get time to really spend time on the analytical methodology or the testing. And always the budget is a major issue when you're working with the early startup or early stage companies. So getting the right equipment is also a challenge. So yeah, it's a good question. Are we really investing enough? But we should be investing. I think that's the question. That's the point I would like to bring. We should be investing enough in our methodologies, right? Finding the right testing site. If you don't want to bring this expertise in-house, get a right testing site, get in the queue, define your CQAs appropriately from the beginning itself, and then monitor them over time. So now we have a great technology available. Trend the data. Don't just ignore the signals. Monitor the data in the real time to see what's happening. And if you're not getting to the answers you're looking for, investigate and try to see how you can make this better. Typical technologies, even in the process, I highly recommend putting the in-process technology in place, like CD Spectra or maybe FTIR or whatever online processes you can put in place. So then you're getting the real-time data versus waiting for the testing in the end. So, and if you see investigations, if you see OOS, investigate, try to understand where is it coming from. Going back on my previous argument, when you're doing the investigation, spend some time to understand why did we have this OOT or OOS versus just, closing the topic and moving forward, because it comes back and haunts you. So I would, let Doug or Hyang think, I'd suggest,
Yeah, I agree with everything that Deepa says. I mean, just because a product like the GLP-1s is in such high demand, it doesn't give us a license to compromise on quality. It's absolutely inexcusable. And certainly at Kylera, we would never, and I don't think any pharmaceutical company would allow us to cut corners on quality just to make sure that we either got a batch out on time or that we got more batches out that were sloppy. There's no compromise here on quality, whether it's during the IPC process when you're looking at in-process checks, whether it's in the final release testing, or whether it's on stability. All three of these things, it's absolutely non-negotiable to have anything but the highest quality possible. And if that means working, as Deepa said early on, to develop very robust methods, you've got to invest the time to do that. There's no other way around it. The last thing I tell my team all the time, the last thing I want to do is have to go to my board of directors or to my executive team, or even worse, to a patient and say, gee, sorry about that batch that we tried to rush through. We ended up injuring a patient, or we ended up having zero efficacy just because we tried to push it through. So we at Kylera, and I'm sure most places elsewhere, are always fully committed to quality. It's a mindset. It's a table stakes assessment that you have to make just to be in this game. We're such a highly regulated industry. You can't compromise. If you try and compromise on quality, you will get caught at one point or another, and the consequences of that are just horrendous and miserable for both the company, but more importantly for the patient. So put the time into quality, make it part of your mindset, and accept it as part of the business that we're in.
Now, I'd love to jump into some of the assays. Oh, sorry, Deepa, go ahead.
No, I was just going to add to that. It's like, I always suggest my, organizations, like when you're developing the product, that's the time to make mistakes. So it's okay to have an OOS at the time versus not having that capturing at the time and not paying attention to the method. And eventually it's going to show up in your final GMP batch, and then you have a lot more investigation to do. So spend the time earlier. It's more forgiving. Take your time, develop it properly, qualify the method, validate the method before you go further. So that's my just 2 cents to that.
Fabulous advice. Now let's jump into some of the assays that we mentioned earlier. I'd love to start with activity. Doug, can you talk a little bit to what assays you're performing for activity and the results that you're seeing from them?
Yeah, so we were fortunate. Our partner, Hengrui in China, had developed bioassays as well. And so we have been able to sort of piggyback on the success that they have to understand the importance of a bioassay. It turns out that there are also commercially available assays that you can get now for the GLP-1s. And so we're doing sort of a combination approach of integrating information that we've already got from our outstanding partner with information that's available at contract test laboratories to make sure that we do have a viable bioassay. like I said earlier, I think the agencies are requiring this now. They're expecting this. So you really can't get away. At least we haven't been able to get away without making sure that that's a very integral part of our overall quality control aspects of our products.
So I will add to that. Typically, bioassays are more variable, but they give you a lot of information, which the other chemical methods, test method don't give. So you're on the right path.
Now, with stability, how often are you running assays on stability? Can you elaborate a little bit there?
Yeah, again, I think these, just because it's a GLP-1 that is deemed as one of these super drug categories, there's no getting around ICH stability guidelines. If you want to be able to have a product that's commercialized globally, it's really wise to go ahead and use an ICH stability program. And then the question is, do you do it on every single batch, or do you do it at various times? Say like every 3 or 4 batches. Well, that really depends on whether you're in the sort of the clinical development phase or you're in the commercial phase. In the early clinical phases, we put as many things on stability as we can so that we can continue to update not only our confidence in our manufacturing process, but also to make sure we can continue to extend shelf life as appropriate. Because as you can imagine, that becomes really critical. In the early days, if you only have 6 months of stability, that makes it very difficult to run clinical trials of any duration. So it's really important for us to continue to collect stability data for those reasons that I just mentioned. And again, we do very standard stability protocols that are all guided by the ICH guidelines.
Now, as you approach scale up, have you seen challenges with purity, potency? Can you talk a little bit about those and how you've overcome them?
Yeah, we've been fortunate as we've scaled up. We haven't seen any adverse effects or adverse impact on either the impurity profile or the stability or the efficacy. I will say that we are in the comparatively early stages, so we're not manufacturing at commercial scale today. But I can tell you that those are problems that you do have to address as you step from one scale to the next, and we'll do that when we get there. I think it's important to recognize, that you already have the Lillys and the Novos of the world manufacturing at commercial scale. So we know that it can be done. We know that we can scale to that level. But I will also emphasize that we always want to have a mindset of continuous improvement. So whatever we're doing at any given time, we'll continue to refine our analytical assays to ensure that we are monitoring maybe the formation of any new impurities that might come up. And whether they're process impurities or whether they're degradants, we'll continue to characterize those things. But the last thing I want to do is get stale and say, well, I developed this assay back in phase 1 and it was fine. I'm just going to take it all the way through to commercial. Because as we know, things can happen at scale up. And that's especially true for the peptides, because there's new issues come in, potentially aggregation of molecules or changes in the secondary structure of these peptides. So we have to be very careful of that. And then, of course, on the small molecule side, we do the same thing. Just make sure that there's no new impurities, no changes in the stability profile. But it's all in that mindset of making sure that we are always trying to get better at what we're doing.
Absolutely.
To add to that, I think it's a part of the development process. When you're doing the scale up, when you have to keep an eye on it, on your processes, and keep monitoring it. So, you target for no impurity, no extra impurity or anything additional, but you still have to keep an eye on it. You can't just close your eyes and assume everything is going to go well.
You mentioned earlier, Deepa, that impurities or issues in the process can be seen real time as an innovation in manufacturing. Can you talk a little bit to that? To those advancements?
What I've seen, correct. So what I've seen people, folks putting the in-process analytics in place as you are moving along through your process. At every step, there are analytical methodology in place. So you can see all this, if there is a trend or if there's a change in your process, you can see it early on versus waiting for towards the end of the process. So that really helps tackle the issues, and also it gives you a better understanding of your process as well, to see what to watch for once you get to the commercial state, because that time you don't have time. You want to get to the market ASAP. So that time you want to reduce the diversions or deviations, so that way if you know your process well enough, early enough, then you can resolve it before you get to the last stage.
And do you see some of that real-time analysis being done with AI in most cases?
Absolutely, yeah. A lot of companies are implementing that. I won't get into detail, but yes, a lot of companies are implementing that.